Hello, everyone. This is Tom Conley from the Physical Oceanography Lab. Welcome to the Thursday seminar. We are going to have a happy hour again at the end of the seminar, and our speaker is going to be able to stick around for a little bit to answer any questions that you might want to ask at the happy hour that you don't get a chance to ask at the end of the talk. Before I introduce today's speaker, I'll go over the rules here. So all audience participants will be muted throughout the seminar. Please do not attempt to turn on your video or share your screen during the talk. There will be an opportunity for you to ask questions live at the end of the talk, just like a normal seminar. Once the speaker has finished their talk, you can use Zoom's raised hand feature to notify me that you'd like to ask the speaker questions. And that raise hand feature is located under the Participants tab at the top of your Zoom window. So now it's my pleasure to introduce Dr. Chrissy Huffard. She's a research specialist in the benthic-pelagic coupling lab at MBARI. She did her PhD thesis on octopus locomotion and behavioral ecology before doing a postdoc in MBARI. And she's also done extensive work in marine conservation in Indonesia. She now focuses on links between deep sea ecology and the surface ocean, and she'll tell us what she's learned from persistent autonomous instruments at MBARI. So welcome, Chrissy.
So thank you everyone for coming today. Thanks, Tom, for the introduction, and thank you, MLML, for the invitation. Today is a really fun opportunity to really share some stories about science and carbon flux and animals living at the abyss based on input from the autonomous instruments at Station M, which is an abyssal time series off the Central California coast. And I'm based at MBARI, the Monterey Bay Aquarium Research Institute, which is a collaboration between scientists, engineers, ship's crew, communicators, people with lots of different skills and areas of expertise. And one of the reasons I really wanted to stress this in this talk is that many of the listeners today are maybe early on in your careers. You may be wondering about the experiences you have and how those experiences might shape or influence your future career options. And I just really want to stress there's no single pathway into science. People come from lots of different experiences. These are the folks in our team. Up on the first two lines, we collaborate with Colleen Durkin, Amanda Kahn, Tom Connolly, Steph Wilson, who are academics externally. Several of you are at MLML. But we have experiences in conservation science, in academia, in industry, and in the military. Our team has really different backgrounds, and we all come together, and those different approaches are really, and experiences are necessary for understanding the ocean on the broad scale, developing technologies to advance ocean science. There's no single perspective that's more or less successful in doing that. It really is a huge team effort. And we work to understand some of the globally relevant questions about ocean science and protecting the ocean. And obviously one of the big ones is what's happening to the ocean and to our Earth in relation to climate change. Since the Industrial Revolution, humans have burned an enormous amount of fossil fuels that has released a tremendous amount of carbon dioxide and other greenhouse gases into the atmosphere. This has warmed the Earth, it has warmed the oceans, causing sea level rise, and lots of human impacts as well. So it's really important to understand this large-scale issue as it relates to the ocean. And this is a video from NASA that shows atmospheric carbon dioxide levels that are modeled over space and time, and warmer colors like red and yellow are areas with higher concentrations of carbon dioxide. Cooler colors, blues and greens, have lower concentrations. And what we can see here is that the emissions of carbon dioxide into the atmosphere are really variable over space and time. There's no blanket one condition that is experienced globally in relation to carbon emissions. And a lot of that carbon impacts the ocean. 90% of the heat that has been, added to our Earth because of our carbon dioxide emissions is taken up by the ocean, and 25% of the carbon dioxide itself is taken up by the ocean. And so that really leads to big picture questions that we're addressing in the lab. How much, if any, of that carbon actually makes its way to the deep sea where it might be stored long term? What happens to that carbon once it's down in the deep sea? And these two main questions really rely on understanding a process called the biological carbon pump, because that's one of the main ways, if not the main way, that carbon makes its way to the deep sea where it might be sequestered long term. So here's a little video of what the biological carbon pump involves.
The biological carbon pump starts when single-celled algae take up carbon dioxide dissolved in surface waters and use it for photosynthesis. A lot of these algae are eaten by small animals, which poop out little carbon-rich pellets. Clumps of these algae, bits of poop, and parts of tiny dead animals make up marine snow. Most marine snow gets eaten as it sinks through the water column, but some of it sinks to the deep sea, where the carbon in it is unlikely to reach the atmosphere again anytime soon. Animals and microbes eat most of the marine snow that reaches the deep sea. This is their main source of food, but a tiny fraction evades consumers, and its carbon can be stored in marine sediments for geologic time.
So this is a process that's happening all over the world. The ocean covers a lot of our Earth, and we study it in the context of the deep sea at Station M, which is a site off the Central California coast. Oh.
Our ocean is vast, covering more than 70% of the planet. Abyssal plains, the flat, muddy, wide-open stretches of the deep ocean floor, cover more than 50% of the Earth's surface and are an important part of the carbon cycle. Station M, 291 kilometers off the coast of Santa Barbara, is 4 kilometers deep, and it's provided an unprecedented window into important abyssal ecosystems for the past 30 years.
So that's a quick window into where Station M is, the habitat that it represents. The abyssal depths, the average ocean depth is 4,000 meters, and yet Station M is one of only just a couple of sites worldwide where carbon flux, carbon supply, carbon dynamics, and ecosystems at that depth have been studied long term. So it's surprisingly unique, although not surprising when you consider the effort of getting down there. Because it is very difficult to reach, the team working at Station M has really focused on developing instruments that can be down at Station M for a long time and persist in that very harsh environment to collect data. And Station M was initiated in 1989 as a time series study in the abyss, and at that time there was it was situated in, it's right about here. If you can see the pointer, it's a little small there, but it sits under surface waters that experience seasonal periods of high chlorophyll concentration. Phytoplankton blooms in the surface waters. And so the question posed by Ken Smith, the person who initiated and really oversees the Station M program, was what happens in the deep sea when there are these changes in the surface waters. And some context here, 50 years ago, one of the prevailing ideas was that the deep sea was so stable that ecosystems down there, animal communities down there didn't really change. And so this idea that even seasonal changes in the deep sea, whether that's changes to the carbon supply or changes to the animal communities down there, it was, pretty new. And so the lab really was advancing that understanding that the deep sea is not a static, unchanging environment. Since the time series was initiated over the years, it's provided evidence that not only does the deep sea relate to seasonal changes, it can actually change based on surface conditions even just a few days later. It also changes with surface conditions and the climate on much longer time scales. So the deep sea is quite dynamic. So the Station M time series relies heavily on instruments called sediment traps, and these are funnels that are strung along a mooring line. At Station M, they're 50 meters above the bottom and 600 meters above bottom in the benthic boundary layer. And they have cups on them underneath. The cups aren't on them in this picture, but those cups rotate. There's a carousel that rotates every 10 to 17 days, and a new cup is under the funnel. 10 to 17 days later, it rotates again, a new cup is under the funnel, and that allows us to get a general time series of what's sinking at any given point. These are cups from a 50 meter above bottom trap. You can see the little dark stuff on the bottom is marine snow. Marine snow is the stuff of the biological carbon pump that sank through the water column. It reached the sediment traps and was collected in these cups. I take them back to the lab, analyze their carbon content and a number of other factors, and that gives us a sense of how much carbon is sinking to the deep sea at any given time in marine snow, or fecal pellets, fish scales, things like that. And that's our understanding of carbon supply to the deep sea. Here's Ken Smith at the top here. He is on top of an instrument called the Free Vehicle Grab Respirometer, which was developed by the lab to study another aspect of carbon dynamics at Station M, the carbon supply, or carbon demand by the seafloor community. If you want to understand where the carbon goes, a big part of that story is understanding how much of it gets consumed. And this instrument was brought out to sea seasonally and lowered to the sea, dropped over the side of the ship, lowered to the seafloor, and it took seasonal measurements of seafloor community respirometry, which is oxygen consumption. Calculations were applied to those data to estimate the carbon demand by the seafloor community. And that gave a seasonal time series of carbon demand, but it became pretty clear that that wasn't enough. The lab needed higher resolution data, and so the benthic rover was developed. This is the benthic rover 2. The benthic rover 1 was developed in the 90s and worked for a while at Station M, but then was lost at sea. This is this benthic rover that has now been working at Station M, not quite for 10 years now, but pretty close at this point. It can handle year-long deployments, and every 2 days moves to a new site, moves 10 meters, lowers the chambers into the seafloor, takes a respirometry measurement for 2 days, and keeps moving. It also takes color images and fluorescence images as well to estimate possible chlorophyll concentration on the seafloor. And just if anybody's curious about the impact of the rover on the seafloor, it's very lightweight. It weighs just a little over 100 pounds. That's spread out over its very large tread, so it barely leaves a mark even in the sediment. This is what it looks like. It's much slower in real life. To save battery, it moves very, very slowly. So this video is sped up many, many times. But this is what it looks like to move. And this is what it looks like when it lowers its respirometry chambers into the sediment. And that gives the picture of carbon demand at Station M to get a sense of who is being involved in these equations, these changes at Station M, at least at the scale that's visible with cameras, there's a time-lapse camera that is deployed as well, and that takes a picture of the seafloor every hour and has been doing so for most deployments since 1989. It's an incredible archive. And the long-term story of carbon at Station M is that for the first 20 years of the Station M time series, more carbon was being consumed by the seafloor community than was being supplied in marine snow. And so this, along with results from other studies around the world, have led to the general understanding that deep sea communities, especially at this depth and this type of ecosystem, are food limited for very long periods of time. This is what that kind of looks like. So we have a time-lapse video here, several months of a sea cucumber crawling around on the seafloor. There's not a whole lot of action. So in several months, we don't really see that many animals. We don't really see that much landing on the seafloor. And it's a pretty quiet scene. And this is fairly representative of the first 20 years of the Station M time series. But then things changed a little less than 10 years ago. These are some sediment trap cups showing a very large amount of marine snow being collected, especially in cup 9 here. So much so that we even had to change the types of cups we used to accommodate how much more material was sinking down. And this really represented a big change in the carbon dynamics at the site. And so if you look at our long-term record of the amount of carbon coming down in marine snow, so POC flux is particulate organic carbon flux. That's the carbon coming down in particulates or marine snow, and over a certain amount of time and area. It's a rate of carbon supply that has really increased significantly over the past few years. You have these very large spikes in the amount of carbon coming down, and what that involves is sometimes years worth, what would otherwise be years worth of carbon coming down in a span of just a few weeks. So if we didn't have sediment traps out there measuring all the time, we could very easily miss one of these events. And these events have an enormous impact on the carbon cycle and carbon budget at Station M. And based on evidence from other sites near coasts throughout the world, it seems like this increase in carbon to the deep sea might be happening on a much larger scale than what we're measuring at Station M. And so if it's true that carbon supply to the deep sea and potentially storage in deep sea is changing, potentially increasing on a large scale, we really need to make sure that's represented in the carbon budget models that are used to understand global carbon cycling. And one of the main ways that people estimate deep sea carbon supply, or the amount of carbon making its way to the deep sea, is through an equation called the Martin model, or the Martin curve. And this is an estimate of how much carbon might be sinking at any given depth based on surface conditions. So surface estimates of chlorophyll production and export. And that might be based on satellite imagery, it might be based on Argo float data, or other measurements from the surface. And so we tested how well the Martin curve and the Martin equation actually applied to Station M based on export flux from the area at that time. And we could see that the Martin curve significantly underestimated the amount of carbon making its way to Station M. In particular, it was completely missing these very important, very quick events delivering large amounts of carbon in a short amount of time. And so how can we better improve these models? Can we model these events? Can we summarize them? And generally, models rely on things being generalized or averaged, and ideally you're generalizing something that's pretty similar across conditions. So we wanted to test whether these large-scale events were all the same or similar and had similar timing and dynamics. And so those sediment trap data are based on 10 to 17 day resolution. And some of the changes that we're looking at could happen over a few days. And so we wanted to use a much higher resolution data set to ask these questions. So we looked at what's called detrital aggregate cover on the seafloor. And detrital aggregates are big clumps of marine snow. So if you picture a large glob of algae, a big chunk of something that sinks all the way to the seafloor, if it's visible in the time-lapse camera, then we can annotate that, make a note of that, and measure how much of that is covering the seafloor within the view of the time-lapse camera. And during the periods that are highlighted in red, those are areas where there was really a lot of gunk or detrital aggregates on the seafloor. And this represents periods where there are very large amounts of carbon landing on the seafloor. But if we trace them back to surface conditions or export flux, that's the amount of carbon estimated to be leaving the surface ocean based on satellite information, we see that it's kind of hard to generalize. Some of the peaks in export flux did translate to peaks in detrital aggregate cover on the seafloor, but some didn't, as we can see from this one. And sometimes conditions on the seafloor signaled a really extreme event, but that wasn't really preceded by anything that we could trace back to the surface. And so by looking at these data sets together, we realized that there's a lot that we're not able to account for, and we're not really close to being able to model these large-scale events based on surface conditions. And that's not entirely a surprise, because the surface export and the landing of this material on the seafloor, those are processes that are separated by 4,000 meters. That's 4,000 meters that's full of fish and other animals, microbes. Some of the events on the surface might have started off with diatom blooms that formed large aggregates. Some of the events in the surface might have started off with much different phytoplankton communities or processed by different zooplankton communities. That midwater column between the surface and the seafloor is full of animals and microbes ready to consume that carbon as it sinks. And we have fish and cephalopods, mollusks, cnidarians, animals of lots of different types that are just a gauntlet of mouths ready for that carbon. They're also repackaging that material into little fecal pellets that can sink down. All of that, whether it's marine snow or those fecal pellets, they can be eaten by microbes on the way down. So it's very easy for a lot of that carbon to be consumed on the way down. And so here's just a beautiful picture of one of those consumers that is a siphonophore that is very importantly linked to a lot of the animals in the midwater. And one of the stars of the carbon sinking show here is the larvation. And this is a small invertebrate that creates a mucus house around it, almost like an extremely elaborate spit bubble that is made out of mucus, that has little chambers in it, and it's extremely efficient at filtering water. These animals are very abundant in the Monterey Bay and along the California coast and they filter an enormous amount of water, and they do that because they're consuming material that's in the water, that's carbon that would have otherwise sunk. So they're large-scale consumers, but they also, when they're done with their mucus house, they shed it. And that mucus house sinks very rapidly, taking all of the carbon in it, sinks it very quickly to the seafloor and brings lots of carbon to the seafloor. And so all of these dynamics, all of these consumers are changing pretty constantly at short and long time scales. And so it's no wonder we see so much variation at the deep sea. You know, if we think about what it takes to go from the surface, where these pyrosomes are, for example, all the way down through the water column, it's a very long path, and that path has different densities of animals and consumers. So that's 1,800 meters, 3,000 meters. I guess the depth's cut off here, sorry about that. And by the time that material reaches the seafloor, it may or may not be in very different condition. The composition of it that lands on the seafloor can be quite variable. And so to illustrate this, I'm going to show you pictures taken by the Benthic rover fluorescence system. The screen is black now, that's not a problem, your computer's not broken. The fluorescence camera images taken by the Benthic rover are completely black if it doesn't detect anything that fluoresces. If it detects something that fluoresces, that shows up as white. And that material may have chlorophyll in it, which signals that it was on the surface not too long ago, and it's still relatively fresh. And if we are able to notice a little bit, the shape and texture of the material that is showing up as being fluorescent is pretty variable. And so it's not just a matter of whether something is fresh. You know, we really have to get into the nitty-gritty of what this material is to understand its impacts on the carbon cycle. And so to get into that nitty-gritty a little bit, the lab developed an instrument called the Sedimentation Event Sensor, the SES, and this is an instrument that is based on the standard sediment trap shape, but instead of having bottles underneath that collect at a 10 to 17 day scale and combine 10 to 17 days worth of data all into one sample, it collects material onto a slide for 2 hours, takes a fluorescence reading to get a general sense of how much chlorophyll might be in it, and then takes a macro image. And from that macro image, we can estimate the light attenuation. So that's gives us a general sense of how much gunk is on there, how much marine snow is settling at any given time. And Colleen Durkin and students have collaborated with us, along with Steph Wilson in the UK, to estimate how much carbon is in that material sinking down. You know, identifiable fecal pellets and aggregates have different levels of carbon, different concentrations of carbon in them. And so really teasing apart exactly what is coming down at very high resolution can give us a sense of the processes and mechanisms and organisms responsible for carbon flux. And this is a daily view of what one of the deployments of the SES has shown us. And so from that, we can see that sometimes, say from March to late April of 2015, you have a whole lot of aggregates bringing carbon to the site. And sometimes, maybe overnight, that story changes. In late May, overnight, all of a sudden, tabular fecal pellets, which are generated by salps, gelatinous zooplankton that live in surface waters, all of a sudden, salp fecal pellets were the dominant supply of carbon to the site. And then as they became less abundant, all of a sudden, cylindrical fecal pellets that might be supplied by fish or crustaceans became the dominant supply of carbon to the site. And so this tells us that on both short and long timescales, there's very high turnover in both the organismal pathways and potentially physical pathways bringing carbon to Station M. And so we have to keep watching over long time periods, but also at very high resolution to understand the turnover processes. And to understand how this relates to the carbon supply and the demand and the carbon budget, we compared the data from the SES to the data from the rover, the carbon demand data, to calculate the carbon budget balance. So was there a surplus or a deficit? If there was a surplus, what was responsible for that surplus? And that comparison showed us that salp fecal pellets, at least temporarily, were the primary source of a carbon budget surplus. It's not, we're not able to say whether there was long-term storage of carbon in the sediments. There might not have been. That carbon might have been eaten over the next 2 years if not much carbon came down, if there was a further deficit in the carbon supply. But it at least helps us understand that if we are going to understand carbon sequestration in marine sediments long-term from biological processes, we really need to understand the organisms that are responsible for that. And in late, I guess it was 2012, there was a large bloom of salps along the US West Coast. There were so many salps that they clogged fishing nets, they clogged power plant intakes. They were really considered a nuisance. But I think what wasn't appreciated at the time, which we were able to learn through the Station M time series, was that this salp bloom was also creating an ecosystem service. For us, they facilitated the drawdown of tremendous amounts of carbon from surface waters, transporting that to the deep sea, and it was also helpful to some of the animals down at depth. As happens with gelatinous zooplankton blooms, at the end of the zooplankton bloom, lots of animals died in surface waters, and those dead salps sank to the seafloor in an identifiable condition. They covered the seafloor at Station M, and they provided a really rich food source for lots of animals. This event was preceded also by a large deposition of phytodetritus or algae detritus that was fresh and potentially full of nutrients, which was also really important because some of these animals actually living 4,000 meters deep rely on nutrients from fresh surface phytoplankton in order to reproduce. So this was a very high quality food source to them. And so what it did is it changed the community from this video that I showed you earlier, where not much was going on, to a situation that looked like this. This is sped up a little bit more, but you can see globs landing on the seafloor and being eaten. And this is a very large phytodetrital event, one of these big carbon pulses. You see all this material landing on the seafloor. It turns the seafloor brownish. The animals consume it, microbes are consuming it, so much so that by the end of the video, it's not really all that brown anymore. So I'll show that one more time. This is a very important event from a carbon flux standpoint and from an animal ecology standpoint, but it's very quick and would be very easy to miss if we didn't have instruments down there all the time. And one of the animals to benefit from this change in the carbon story at Station M, this recent increase in the amount of carbon coming down, is a little sea cucumber called Peniagone species A. And here we have 3 size classes, small, medium, large. We did not place them there, it was just a lucky shot. And they really seem to take advantage of this food resource. They eat fresh phytodetritus that lands on the seafloor and have been very good at taking advantage of it. So if we look at a seasonal view or an annual view, which is what, if we only were able to look at the data we measure when we just go to sea, then this is the view we get of how Peniagone's population density, as revealed by ROV surveys, changed after the large food flux events in 2011. The population densities were very low before and they just skyrocketed after, especially in late 2014. And if we didn't leave instruments on the seafloor at Station M, this is the view that we would have. But we do leave instruments on the seafloor at Station M that have given us a daily view. These are data from Larissa Lemon's CSUMB master's thesis and a paper that she's probably about to submit any day now. And from a daily view, we can see that the densities increased and decreased over the course of several years. And this was a very long event. By coupling it with size information, we can tell when immigration and migration might have happened. And this is a really helpful view to have. Going further, we even have an hourly view from the time-lapse camera. And if we look at the hourly view in leading up to the peak in densities, we can see that there's a large number of individuals herding on the sea floor, about in the same direction in relation to the current. And these large scale dents happened to be in just the right spot when the currents changed, and you can see large chunks, globs of stuff landing on the seafloor. That's fresh phytodetritus, that's fresh food. Then a benthic storm comes in and stirs everything up. That's happening now. And they eat that, and then they change direction. And these are very important events ecologically speaking. It's a huge source of food to these animals. The fact that we think they can migrate in large numbers is ecologically relevant. And this is a view that we would not have had if we only went out once a year. And this is just for fun to show you. Penny agony also happens to be able to swim. And so it can consume carbon from one area, and it crawls quite slowly, but it can move much faster if it drifts in the water and in the currents. And so this individual is swimming, and oh, I guess there was sound on that. Didn't realize. I hope that wasn't loud for you guys. This individual is swimming and will be able to move its carbon to somewhere else. Now that carbon happened to be beneficial to penny agony species A. Not all animals reacted in the same way to these changes in carbon supply. I'm going to show you a video of a sponge, which might seem a little strange, might seem anticlimactic, but you might have seen this before. Sponges are sedentary. They can't move in relation to the carbon coming down. If there's a whole lot of marine snow or salp carcasses coming down, they're going to get rained on. And so sponges' densities declined rapidly after the salp deposition event. After all those salp carcasses landed on the seafloor, they're starting to come back. And it may be in part because of a behavior that sponges actually have that was found by Amanda Kahn at your very own MLML. And when we look at the behavior of the sponges at an hourly scale, but sped up, we can see that they're able to contract. And if you look very closely, it's possible that they might be clearing gunk from their cells in their bodies. And so by having a persistent view on the sea floor, by watching these animals at their pace and adjusting that to our pace, we're able to really see some interesting behaviors that we wouldn't necessarily otherwise see if we were just taking quick snapshots. So in summary, the deep sea is very dynamic, and we have that understanding of these dynamics because we've observed it over very long time periods, but also at a wide variety of time resolutions, and different approaches to observing it. Deep sea carbon flux and storage depends on a lot of different processes that take place all the way from the surface to the sea floor, and so modeling them will be a very complex process that requires uninterrupted long-term observations, ideally at high temporal resolution and throughout the water column. These are needed to understand carbon dynamics in the deep sea. And so our next steps are to really continue to piece these things together, but also respond to changes as they come along and gain attention and relevance. So many people think of a time series as just always measuring the same thing all the time, and that's not necessarily the case. We change our questions as the research evolves. And one of the biggest questions that we've had recently, even before these recent events of wildfires along our coast, is how does wildfire relate to the carbon dynamics at Station M? So our large increase in carbon flux at Station M also corresponds with a large increase in wildfires along our coast and inland along the West Coast. As we've seen and experienced, and I hope everybody listening here is has a safe airspace to breathe and has a safe home, hasn't lost their homes from these wildfire events, we can see that those events have not just impacted us on land, they're also blowing smoke out to sea. And that smoke can settle, and that smoke carries a lot of carbon in it. During these events, a lot of not just small pieces of ash, but large pieces of charred material, twigs and needles and entire, leaves of trees burned and charred were making their way to the ocean. And that created a soup in the water that's washing up on beaches, but also very likely settling to the deep sea. And so we're really looking forward to going back out in October to see if any of this material has reached Station M yet and followed over time, because it might take a while to get there. So that's what we're up to next. So thank you very much for attending and listening. Thank you to the David and Lucille Packard Foundation for funding all of this work, the crew of the Western Flyer for supporting our shipboard operations, and animations in this video were created by Eric Knight Maps and Frame 48. And please do follow us on social media. We're at MBARI News. We're on Facebook, Twitter, Instagram, and YouTube. So thank you very much.
Thank you, Chrissy. And now we're happy to take any questions. You can use the raise hand feature, which is under the participants tab in Zoom. And so while we're waiting for questions, actually, I have a question about, this Station M is, it's in the California current system, which is highly productive and might not be necessarily, of course, not representative of all deep sea ecosystems.
Yeah.
How far do you think you'd have to go before you would need another station to understand the ecosystem?
Well, in a dream world, I'd have a large transect from California to Hawaii, but we don't have that. There is Station Aloha off of Hawaii that has data to show that the carbon flux there and at other open ocean sites might be decreasing because there's surface warming and changes in phytoplankton communities might be limiting mixing in carbon flux. How far that coastal influence extends, I don't know. I think we might get a sense from the satellite imagery to see how far those phytoplankton plumes can make their way out. But, I don't know. That's a great question. I think it would be highly variable from year to year.
Okay, and just a follow-up on that. Are there, besides Station M, and you mentioned Station Aloha, how many other sites, really intensively studying the seafloor over long times are there? Is there anywhere else you can, even compare your data with at this point?
Yeah, our main comparison is from the Porcupine Abyssal Plain, which is a time series that's older than the Station M time series in the North Atlantic. That's run by various groups in the UK, mainly NOC at University of Southampton, in conjunction with University of Southampton. They don't collect the exact same data sets that we do. They don't have a time series of carbon consumption yet. I think that is something they would like to have, but they also have time series that we don't have. They have a different picture of animal reproduction and nutrient use by the animals that we don't have. And so even though they're not collecting the same things, it's close enough to get a general sense that their carbon dynamics are also changing. They have coastal influence as well, so they're seeing an increase in carbon flux to their sites. There are other sites that are emerging, and so maybe they're not quite as long running, but they certainly have the potential to be and are collecting very interesting information. The Delos site, and throughout the world there are several, but they're just not as long running as the Station M and PAP sites and Station Aloha. BATS, in Bermuda, the Bermuda Atlantic Time Series has sediment traps that have been running for a long time and have given a general sense of, like Station Aloha, what's happening in more oligotrophic situations.
Okay, thank you. And it looks like we do have some questions coming in with raised hands, and Annie Bodell had her hand up first. Annie, I will unmute you.
Hi, Chrissy. Great talk. Just following up to Tom's question about kind of those other deep time series, I was really intrigued that when you had the temporal resolution that Station M had, that it really kind of contradicted the Martin curve's expectations for that deep flux. Do you know if that is happening at some of those other abyssal sites in terms of underestimation, or do they not have the same temporal resolution that Station M has?
So luckily the Martin curve, in order to make that comparison, you need POC flux from depth, which is available at other sites like the Porcupine Abyssal Plain, and surface chlorophyll or surface estimates of export flux. And that is something that's widely available. And it seems like the Martin curve does underestimate these events. Interestingly, at other sites, interestingly, when these events aren't happening, it's very, very good at estimating flux to depth. And so, it's not really a surprise that the Martin curve wasn't developed for this purpose. The Martin curve stems from a paper that was describing carbon flux at different depths. That curve was then taken and used for lots of different other uses, including estimating carbon flux at depth, but it wasn't designed for that. And so it's not really a surprise that it will need to be tweaked in order to accommodate things like this.
Right, that makes sense. Thanks.
Okay. And we have another question from Travis. Travis, I'll unmute you now.
All right. Really, really interesting talk. It's quite interesting. I was curious. I'm a little afraid to try and pronounce that sea cucumber's name.
Don't worry about it. Just say the sea cucumber.
Right. So I was curious if you saw, along with an increase in the density of that sea cucumber, if there are any other predators that prey on that sea cucumber that aggregated, or any other species.
Yeah, we did not. And so the trophic links at Station M are, not super complex because most animals are deposit feeders. There are some predators and scavengers, but most of them are very small. And so would be consuming maybe the larval recruits or smaller life stages that we're not seeing. There are some larger fish in the area. Rat tails are quite abundant in the area, and they have become more abundant in the area based on results from a thesis by Danielle Fabian, who just finished up at CSUMB. But we didn't see any predation events, and it doesn't seem like any of the fish that we know of are large-scale predators of those sea cucumbers. And so it's an interesting story to understand these trophic dynamics that are, I don't want to say they're predator free, but they have much less predation pressure than a lot of the systems that are typically used to describe ecological theories in relation to feeding.
Very interesting. Thank you very much.
Thanks.
Hey, thank you for your question, Travis. And Logan had a question next. And you're unmuted, Logan.
All right. Thank you. Thank you so much for the talk. That was really interesting.
Thanks.
Kind of going off Travis's thing about the sea cucumber density, I was wondering, or I noticed it, the increase in density kind of coincided around the 2015 to 2014 time range. And I was wondering how much of that, if you know, was due to the big array kind of close.
You cut out quick in the middle of that. Can you please repeat after you started mentioning 2015?
Oh, yeah. Is my mic all good now?
Yeah.
Sweet. Yeah, so I was wondering how much of the density increase for the sea cucumber was due to potential phytoplankton falling from the phytoplankton bloom events that happened at Monterey.
So I think that, so the increase in, we didn't see an increase in 2014 and 15 at Station M based on conditions at Monterey. They're pretty far from each other. And it seems like, I don't know how wide ranging any blooms in 2014 and 15 in Monterey were, but it doesn't seem like most of them reached Station M. And in general, that year was actually pretty depauperate. There wasn't really much coming through that year. And, you know, those were blob years where the surface in general offshore were really, really starved and there wasn't a lot going on in the surface. The currents were also really, really strange. And so in general, until we keep watching for another 50 years and see more of these events, it's hard to generalize how they're related.
Gotcha. Thank you so much.
Oh, looks like I'm unmuted already.
Hi, Amanda.
Hi, Chrissy. Cool. I saw the sponges. That was great. Thank you for that. But my question is actually about plankton blooms. So I know that you have such great resolution to even see seasonal changes in things. Is there any difference between the spring bloom and the kind of fall phytoplankton blooms and the productivity that results? Is there any kind of patterning to that on the seafloor, or because the zooplankton can really vary with who blooms up, does that kind of go away?
So in terms of the seasonality of the amount of carbon making its way down, there is some seasonality to that if we look at the entire time series, mid to late summer, there are peaks, but that seasonality has really been breaking down in the past 10 years. If we compare the seasonality of the first 20 years of the time series to the last 10 years, it's very different. So I think that is something that is related to changes, in all sorts of, phytoplankton dynamics. In terms of seasonality of what's actually coming down, I think Colleen is actually the best person to answer that based on students that she's worked with who have looked at the sediment trap archive. We use the sediment traps to measure carbon, but we also keep a sample for microscopy. And Colleen has worked with 2 students, including Cindy at MLML, to understand potential seasonality and changes in the phytoplankton community over time. And I think Colleen would be better suited to answer that if she's listening.
Yes, great. Thank you. And
thanks, Amanda.
Okay, and we also have a question from Jim Harvey. And you're unmuted now, Jim.
All right, thanks, Tom. Nice to meet you, Chrissy. Great talk.
Thanks. Nice to meet you too.
You may have already sort of alluded to this, but I guess my question is about these large flux events that occur every once in a while. And maybe you don't have enough data yet, but you kind of implied that probably it's these big events that really supply enough energy to the deep sea to be able to keep the community going. Do you have enough data to show that it's really kind of dependent upon those big events occurring every once in a while for them to kind of keep the community going?
Well, it has to come from somewhere, right? So, you know, I can't run my bank account in a deficit for 20 years unless I started off with something in there. And so, you know, it's possible that there was clearly some large supply of carbon to station M before the time series started, enough that it was able to run that deficit for 20 years. And how much these large events actually account for all of that surplus, you know, all of that carbon storage in the sediment, we can't exactly say. It makes sense that these events happening when they do, maybe they happen on maybe they're new now, or maybe they happen on a 20, 30, 50-year cycle. That we can't answer without much longer term monitoring. But it's clear that they're part of the picture, even if we can't say whether they're the entire picture. There are other aspects of carbon supply that these animals could be feeding off of, you know, our estimates of the supply, our comparisons of supply and demand don't take dissolved carbon and bacterial activity into consideration, and so that is part of the story as well. But this general view that we have suggests that these very rapid and impactful events could be supplying many years worth of consumption.
Right. So yeah, and I guess the follow-up then is, how confident are you guys with the respiration data? Are you sure that that's sort of realistic of what they're consuming?
Yeah, so the respiration data, there are certain aspects that we are working to improve. Our calculations of carbon consumption depend on a respiratory quotient that does not change in our calculations based on conditions. We know that respiratory quotients do change based on conditions. We just haven't been able to incorporate those changes. And so we are actually working on improving our sampling system so that we get a better understanding of variation in respiratory quotients, so that we can adjust those calculations accordingly. And if that's something we can generalize, then we might be able to go back into the time series and recalculate things to see if that story changes.
Great, thanks very much.
Thank you very much.